Structure in Unit 1. In this section, we will finally be moving on to living cells. Our cells are surrounded by a membrane called the plasma membrane. Inside the cell, the contents are called the cytoplasm. Within the cytoplasm, we have organelles, which are specialized structures. The cytosol is the fluid part of the cell, not to be confused with the cytoplasm, which is all of the contents on the inside. Somewhere near the center of the cell, we have the nucleus, which will contain the genetic information. Here we have an illustration of a cell. Keep in mind this is an artist's representation of what a cell would look like. If you were to view a cell under a light microscope, what you would typically see is a somewhat round object with a dark area near the center. The plasma membrane plays a very important role in the cell. It is almost as important as the genetic information. It is composed of a lipid bilayer made up of two layers of phospholipids. Inside is hydrophobic and outside is hydrophilic. It also has selective permeability, which is going to allow certain substances to pass into the cell while others cannot. This will allow certain things to come into the cell freely, such as steroid hormones, while other substances will not be able to cross the membrane except under special circumstances. The plasma membrane will also play a role in identification of a cell. Cells will have markers to identify which type of cell they are and to communicate with other cells. Some examples of these markers are markers to identify your cell as human, markers to identify fat cells and where to store body fat, markers to identify cells as you as an individual. We have different ways things can cross in and out of a cell by crossing a cell membrane. Passive or simple diffusion is going to be the simplest method. No energy is required, and the substance can cross anywhere on the cell membrane. To give the cell a little bit more control, it can also use passive transport. Here, no energy is required either, but the substance must cross at a specific location. Often these locations are carrier proteins that are going to cross the cell membranes. This can be compared to using a bridge to get it on and off of an island. With active transport, energy is going to be required. This is going to offer the most control, and the substance must also cross at a specific location. This can be compared to a toll bridge to get on and off of an island. It will cost the cell in order to move the substance. Fluid balance in the cell is going to be largely controlled by the concentration of various substances. The concentration is the amount of a given substance in a particular volume of fluid when looking at the cell. The osmotic pressure is going to be the pressure of water. Water and the solid substances are both going to want to be in the same concentration on both sides of a membrane, and will oftentimes be willing to do work in order to be in equal concentrations on both sides. If something is hypertonic, it refers to it being a higher concentration. If something is hypotonic, we refer to it being a lower concentration, and isotonic refers to it being the same concentration on both sides of the membrane. You can follow the link below to see a visual demonstration of how the fluid balance works with osmotic pressure.
This membrane separates pure water on the left from a solution of sucrose and water on the right. The membrane is semi-permeable. Water can cross it, but sucrose is too large for the membrane's pores. Water molecules will move from a solution with a lower concentration of solutes to one with a higher concentration of solutes. A solution with fewer solutes has more water molecules that are free to move about and follow their concentration gradient across a membrane. Thus, there will be net movement of water across this membrane from left to right. This diffusion of water across a membrane is called osmosis. Osmosis is of great importance in the water balance of all cells.
Crenated cells are an example of what happens when you have a difference in osmotic pressure. Crenated cells are just dehydrated cells. If you were to take a red blood cell and place it on a plate, there is a higher concentration of water inside the cell than there is outside of it. The water will move from inside the cell to outside, and the cell will become dehydrated, taking on a spiky type of appearance. On the other hand, if you were to take that red blood cell and place it into water, there would be a greater concentration of water on the outside of the cell than inside. Water would cross the membrane and start to cause the cell to swell and eventually burst. This is why we have a mixture of solid substances in an IV rather than just plain water. Some substances are too large to use active or passive transport. When these substances are too large, the cell has another means to get them in. One of them is endocytosis, which means the cell is taking something in or engulfing something. When there is a solid particle, we refer to it as phagocytosis, which literally translates to cell eating. Penocytosis means cell drinking. In both of these cases, the cell will wrap itself around the particle and engulf it. Penocytosis is a term you will probably not see used outside of the textbook, although you will see endocytosis and phagocytosis used frequently. Exocytosis is the reverse process, where the cell removes something or excretes something. Oftentimes it will do this through a vesicle, which is a membrane surrounding a substance produced by the cell. The reason for using a vesicle, if you are creating an enzyme that can damage the cells, you would want to surround it in a vesicle or a membrane to protect the cell that has created the substance so that it is not harmed. Here we have an animation produced by McGraw-Hill.
The substances taken in by single-celled organisms are often particles or large polar molecules that cannot cross the hydrophobic plasma membrane. Many single-celled eukaryotes employ endocytosis to ingest such food particles. In this process, the plasma membrane extends outward and surrounds the food particle. Cells use three major types of endocytosis: phagocytosis, penocytosis, and receptor-mediated endocytosis. If the material the cell takes in is particulate, such as a bacterium or a fragment of organic matter, the process is called phagocytosis. If the material the cell takes in is liquid, it is called penocytosis. Specific molecules, such as low-density lipoproteins, LDL, are often transported into eukaryotic cells through receptor-mediated endocytosis. Molecules to be transported first bind to specific receptors on the plasma membrane. The interior portion of the receptor protein is embedded in the membrane. The protein cladrine coats the inside of the membrane in the area of the pit. When an appropriate collection of molecules gathers in the coated pit, the pit deepens and seals off to form a coated vesicle, which carries the molecules into the cell. Exocytosis is the reverse of endocytosis. This process results in the discharge of material from vesicles at the cell surface to the outside of the cell.
The fluid inside the cell is the same thing as the interstitial fluid, which is the fluid that bathes the tissue. If you were to cut open a body and look inside, even without blood, you would notice things are moist and wet because of the interstitial fluid. It is largely water, 75 to 90%. It will also contain glucose, amino acids, lipids, ATP, and waste products. The cytoskeleton will function similar to the way your skeleton does. It helps to provide support and structure to the body, as well as roadways within the cell for moving products. There are different types of fibers and filaments that make up the cytoskeleton. The microfilaments are good for giving shape and strength. They will also provide support and movement. They will create little finger-like extensions or projections called microvilli. The intermediate filaments are slightly larger and are used to hold organelles in place. They will also be used in places where you have stretching or tension, such as where you create a pseudopodia to engulf a substance. The microtubules are the largest of these fibers and help provide the cell shape, but also help with the movement of organelles, making cilia, flagella, and moving secretory vesicles inside the cell.